Hi, Dr. Biology here, and this is about the formation of tissue fluid, and it's for A-level biology. Three, two, one. Hi everybody, it's Dr. Biology here, and this is the formation of tissue fluid, and it's part of A-level biology. So firstly, let's have a look at this diagram. So quite a complicated diagram. I'm going to try and explain it to you. So here we go. So at the top, what we have here is the blood coming in from the arteries and the arterioles. And you can see that within the capillary, you've got things like you've got water molecules, and then obviously you've got oxygen, nutrients, and other amino acids. So you've got oxygen inside the red blood cells that comes out. Okay, and then you've got red blood cells, and these are plasma proteins, and they're all dissolved within the plasma. So what we can see here is at this end there is a higher hydrostatic pressure than it is at this end, at the venial end. Osmosis, the osmotic pressure is exactly the same all the way across the capillary. So I'm going to try and explain, well, how is tissue fluid formed? So here we've got the tissue fluid being formed and going into the cells, and then excess tissue fluid going to the lymph vessels. So we're going to talk about how that's formed, then how some of the tissue fluid, particular water molecules, can then go back into the venial end of the capillary. So first of all, as you can see, hydrostatic pressure is greater in the capillary at the arterial end. So there's a greater hydrostatic pressure. So therefore the overall outward pressure forcing the fluid out of the capillaries and into the spaces around the cells forming tissue fluid. And that's shown by the yellow arrow. This includes things like oxygen, nutrients, amino acids and water. But you'll notice that the plasma proteins, such as red blood cells, antibodies and other small proteins, remain in the capillary. And then at the venial end, what we've got here is at the venial end, hydrostatic pressure reduces in the capillaries. So you can see there's a smaller amount of hydrostatic pressure, as shown by this smaller yellow arrow. And then we can see that as the pressure reduces in the capillaries, there's fluid loss, and so inside the capillary there's a higher concentration or increase in concentration of plasma proteins, and because they stay in the capillary. Therefore, the water potential inside the capillary, so the water potential at this point in here is much lower than it is outside. Okay, so there's lots of molecules outside, lots of molecules of water. And then this means that some water can re-enter via osmosis. Okay, so it enters the venial end by osmosis, and then any excess tissue fluid that's between the cells then is drained into the lymphatic system and moved through to the blood at the vena cava. So this summarizes the whole thing there. Okay, so you've got to remember two main things. It's about hydrostatic pressure and water potential, and this allows osmosis. So at the arterial end, hydrostatic pressure has a greater effect, whereas at the venial end you've got osmosis occurring because it's a difference in water potential. So here's some exam practice. So what I suggest you do is you have a go at these questions. I think there is one stray question on hemoglobin, but that won't harm you. And then if you go to the end and have a look at the mark schemes and see how you got on. So here you go. So here are some exam questions. I hope you found that useful. There's going to be a lot more coming on A-level biology. So this is tissue fluid. This is paper 1. It could come up in paper 3 as well. Hope they found that useful. I'll see you soon.